Viewing specimen using the light microscope. The light microscope has two lenses. There is the eyepiece lens and the objective lenses. Now, if you look at a diagram of the microscope, it's that there are different parts, each part playing a very important role in ensuring the viewing of the specimen. There are two knobs that bring about adjustment and focus of the specimen. There is a coarse adjustment knob that moves the body tube up and down to bring the specimen into focus. Fine adjustment knob also moves the body tube up and down, but over very small distances, and in that way it facilitates the focusing of the specimen in such a way that it becomes very sharply focused. The coarse adjustment knob moves the body tube up and down over long distances, and therefore it brings the specimen into rough focus. But to fine-tune the view of the specimen, the fine adjustment knob is used because it moves the body tube only over small distances, and this enables the sharp focusing of the specimen that is mounted and is being observed under the microscope. Now, there is a revolving nose. This is a revolving nose that can be turned, and in so doing it changes from one objective lens to the next. So by moving the revolving nose, one is able to shift from one objective lens to the other. Now, each of these two types of lenses has its own magnification. In each microscope, the eyepiece magnification is constant, it does not change. So for example, if it is times 5 or times 10, that magnification will remain constant. On the other hand, there are three objective lenses depending on the magnification. That is, there is a low power objective lens, medium power objective lens, and high power objective lens. And only one objective lens can be used at a time, because only one can fit into the slot immediately below the eyepiece lens. So using the revolving nose, one can shift from the low power to medium power to high power depending on the requirement of the viewing. Now, magnification. When it comes to the magnification, remember that there are two lenses that are used to view the specimen. There is the eyepiece and one of the three objective lenses. So to calculate the total magnification, it will be therefore necessary to multiply the magnifications of the two lenses, and this is obtained from this formula. You multiply the eyepiece lens magnification times the objective lens magnification. For example, if the eyepiece lens has a magnification of times 10 and the objective lens in use also has a magnification of times 10, hence the total magnification is obtained by multiplying 10 times 10, which gives you a total magnification of 100. So the object that is viewed under the microscope will appear 100 times larger. The specimen is placed at the stage of the microscope, and light rays coming from source, whatever the source that is, comes and hits the mirror. The mirror reflects the light rays to the condenser. The condenser concentrates the light rays into very powerful beam. Then the light rays pass through the specimen on the stage, illuminating it. Light rays pass through the specimen, then through the objective lens. The objective lens will now refract the light rays to provide the first level of magnification. The light rays travel through to the eyepiece lens, where the second magnification takes place. So eventually, when observing at this point here, the image that will be observed will be multiplied, will be several times larger than the object at the stage. The first magnification is carried out by the objective lens, then the second magnification by the eyepiece lens, so that the eye views the image of the specimen on the stage that is many times larger, and that is how the magnification comes about. So the total magnification is calculated by multiplying the two magnifications of the two lenses that magnify the image of the object. Now, the specimen that is under observation is not placed directly on the stage, but instead microscope slides are used. These are thin, rectangular, transparent glasses onto which the specimen to be viewed is placed and then mounted on the stage of the microscope. So this is a microscope slide. Specimen is placed on the slide, and usually a very thin glass cover slip is placed on the specimen. This is mainly to protect the objective lenses from damage and staining. So this slide is then placed on the stage, and it is held in position. It is fastened in position by clip. There's a clip on the stage that holds the microscope slide in position to prevent it from slipping off and falling off the microscope.